गाइस, आई एम आर शर्मा और आप लोग देख रहे हैं BSCG दिसंबर के फुल सॉल्यूशन वीडियो। तो स्टे ट्यून।
1a. Describe the DNA double helix structure. Solution. The DNA double helix is a twisted ladder-like structure made of two strands that coil around each other. The strands are composed of nucleotides, each containing a sugar phosphate backbone and nitrogenous bases, adenine, thymine, cytosine, and guanine, that pair together, A with T and C with G. This pairing forms the rungs of the helical ladder, creating a stable structure crucial for genetic information storage and replication. The strands twist around each other in a right-handed spiral, forming the iconic double helix. This configuration not only securely stores genetic information but also allows for efficient replication. The DNA double helix is vital in the transmission of hereditary traits from one generation to the next and serves as a template for the synthesis of proteins, playing a crucial role in the functioning and development of all living organisms. B. What are the forces that account for the stability of the DNA double helix? Solution. The stability of the DNA double helix stems from a combination of molecular forces acting within its structure. At the heart of this stability lie hydrogen bonding and base stacking interactions. Hydrogen bonds form between specific pairs of nitrogenous bases across the two DNA strands. Adenine, A, pairs with thymine, T, through two hydrogen bonds, while guanine, G, pairs with cytosine, C, through three hydrogen bonds. These hydrogen bonds serve as the rungs of the DNA ladder, holding the complementary base pairs together. This specific base pairing is fundamental for maintaining the genetic code and facilitating the accurate replication of DNA. In addition to hydrogen bonding, base stacking interactions play a critical role. Adjacent bases within the same DNA strand align and stack atop one another in a manner that optimizes the overlap of their electron clouds. This stacking interaction involves the aromatic rings of the bases, allowing for van der Waals forces to stabilize the helix structure. This stacking also minimizes exposure of the hydrophobic bases to the surrounding aqueous environment, contributing significantly to the overall stability of the DNA molecule. Furthermore, the helical structure itself contributes to DNA stability. The double helix twists in a right-handed manner, which aids in distributing mechanical stress, ensuring the molecule's resilience during various cellular processes like replication and transcription. These combined molecular forces, hydrogen bonding between complementary base pairs, stacking interactions between adjacent bases, and the helical structure, synergistically contribute to the remarkable stability of the DNA double helix. This stability is pivotal for the accurate storage and transmission of genetic information, serving as the cornerstone of life's genetic blueprint in all living organisms. 2a. What are eicosanoids? Explain their physiological functions. Solution. Eicosanoids are a class of signaling molecules derived from polyunsaturated fatty acids, primarily arachidonic acid. These bioactive compounds play pivotal roles in regulating various physiological functions within the body. Prostaglandins, one type of eicosanoid, are involved in inflammation, vasodilation, and the modulation of blood clotting. They mediate pain and fever responses and contribute to immune system regulation. Thromboxins, another subgroup, promote blood clotting by inducing platelet aggregation and vasoconstriction. Leukotrienes, also derived from arachidonic acid, play a crucial role in immune responses, particularly in inflammation and allergic reactions. Eicosanoids act as local hormones, exerting their effects in proximity to their site of synthesis. Their rapid and transient nature allows for precise and localized responses, contributing to the body's ability to maintain homeostasis. Despite their involvement in normal physiological functions, this regulation of eicosanoid pathways can lead to various disorders, such as chronic inflammation and cardiovascular diseases. Understanding the intricate balance of eicosanoid signaling is crucial for developing therapeutic interventions targeting these pathways. B. Differentiate between reducing and non-reducing sugar with suitable example. Solution. Reducing and non-reducing sugars differ in their ability to donate electrons, act as reducing agents in chemical reactions. Reducing sugars have a free aldehyde or ketone functional group that can undergo oxidation, reducing other substances in the process. Examples include glucose and fructose. In the Benedict's or Fehling's test, reducing sugars reduce copper ions, forming a precipitate. Non-reducing sugars lack a free aldehyde or ketone group, so they cannot act as reducing agents. Sucrose is a classic example of a non-reducing sugar. It consists of glucose and fructose units linked by a glycosidic bond, effectively concealing the reducing groups. To test for non-reducing sugars, one must first hydrolyze them into their constituent monosaccharides using acid, breaking the glycosidic bond. The resulting solution can then be tested for reducing sugars using standard methods. In summary, the presence of a free aldehyde or ketone group distinguishes reducing sugars, which can directly reduce other substances, from non-reducing sugars, which need to be hydrolyzed to expose such groups before exhibiting reducing properties. 3. Write short notes on any two of the following. A. Gluconeogenesis. Solution. Gluconeogenesis is a vital metabolic pathway that enables the synthesis of glucose from non-carbohydrate precursors. Primarily occurring in the liver and kidneys, it becomes especially crucial during fasting or low-carbohydrate states to maintain blood glucose levels. The process involves converting substrates like lactate, pyruvate, and amino acids into glucose through a series of enzymatic reactions. This metabolic flexibility ensures a continuous supply of glucose for organs, particularly the brain, which heavily relies on this energy source. Gluconeogenesis is finely regulated, balancing the body's need for glucose with the availability of substrates. While glycolysis breaks down glucose for energy, gluconeogenesis complements this by producing glucose when dietary sources are insufficient, contributing significantly to overall glucose homeostasis. B. Electron transport chain. Solution. The electron transport chain, etc., is a pivotal component of cellular respiration, taking place in the inner mitochondrial membrane of eukaryotic cells. Comprising a series of protein complexes, including NADS dehydrogenase, cytochrome BC1 complex, cytochrome C, and ATP synthase, the etc. orchestrates the flow of electrons, generating a proton gradient across the membrane. As electrons move through the chain, energy is released and utilized to actively pump protons into the intermembrane space. This proton gradient serves as a potent driving force for ATP synthesis through oxidative phosphorylation. The concluding step involves electrons combining with molecular oxygen to form water. The electron transport chain is a critical energy-producing process, allowing cells to harness the energy stored in nutrients and convert it into adenosine triphosphate, ATP, the primary energy currency for cellular activities. C. Ketone body. Solution. Ketone bodies are organic compounds produced in the liver during periods of low carbohydrate availability or fasting. The three main ketone bodies are acetoacetate, beta-hydroxybutyrate, and acetone. When glucose levels are insufficient for energy demands, the body shifts to utilizing fatty acids for fuel, leading to the production of ketone bodies through ketogenesis. These water-soluble molecules serve as an alternative energy source, particularly vital for tissues like the brain and muscle during times of glucose scarcity. Ketone bodies can cross the blood-brain barrier, providing
neurons with an efficient energy substrate. This metabolic adaptation is prominent in fasting, low-carbohydrate diets, or conditions like diabetes. While a valuable energy source, excessive ketone body production can lead to ketoacidosis, a potentially serious complication. Understanding ketone body metabolism is crucial for appreciating the body's adaptive responses to varying nutrient availability. 4a. Describe the method for fractionation of subcellular organelles. Solution. Sure, here's a more detailed explanation of the method for fractionation of subcellular organelles. 1. Cell homogenization. Begin by gently breaking open the cells to release their contents. This is typically done through mechanical methods such as grinding or using a homogenizer. The resulting mixture is called a homogenate, containing cell debris, cytoplasm, and organelles. 2. Differential centrifugation. The homogenate is subjected to a series of centrifugation steps at progressively increasing speeds. The first low-speed spin separates larger components like nuclei and unbroken cells, forming a pellet. The supernatant is then subjected to higher speeds in subsequent centrifugations to isolate smaller organelles. 3. Pellet resuspension. Each pellet obtained from the centrifugation steps can be resuspended in a buffer to create a fraction enriched in a specific organelle. 4. Optional washing steps. Depending on the purity needed, washing steps may be included to remove contaminants. This involves resuspending the pellet in a buffer and repeating the centrifugation. 5. Density gradient centrifugation, optional. For further purification, the isolated organelle fractions can be layered on top of a density gradient medium, such as sucrose or iodixanol. During centrifugation, organelles migrate through the gradient until reaching their equilibrium position based on density, allowing for separation. 6. Fraction collection. Fractions containing the isolated organelles are collected from the gradient. These fractions represent different purity levels of the targeted organelle. 7. Analysis and confirmation. Analyze the fractions using various techniques such as electron microscopy, enzymatic assays, or immunoblotting to confirm the presence and purity of specific organelles. By employing these techniques, researchers can obtain purified fractions of subcellular organelles, enabling detailed study and analysis of their structure and function. B. Explain the role of Golgi bodies in protein processing with the help of a diagram. Solution. The Golgi apparatus plays a crucial role in protein processing within eukaryotic cells. Here's an overview of its functions. 1. Modification of proteins. Newly synthesized proteins, after being synthesized in the endoplasmic reticulum (ER), are transported to the Golgi apparatus. Within the Golgi, proteins undergo various post-translational modifications. This includes adding or modifying carbohydrate chains, glycosylation, which is crucial for protein stability, recognition, and functionality. 2. Sorting and packaging. The Golgi apparatus sorts proteins based on their destination. Different regions of the Golgi are involved in processing proteins for secretion, incorporation into lysosomes, or insertion into the cell membrane. Proteins are packaged into vesicles that bud off from the Golgi and are transported to their specific cellular locations. 3. Formation of lysosomes. The Golgi apparatus is involved in the formation of lysosomes, cellular organelles containing enzymes responsible for intracellular digestion. Enzymes are modified and activated within the Golgi before being packaged into lysosomes for their digestive functions. 4. Protein glycosylation. Glycosylation in the Golgi involves adding sugar molecules to proteins. This modification is critical for protein stability, recognition, and interaction with other molecules. Glycosylation also contributes to the diversity of cell surface proteins and aids in cell-cell communication. 5. Protein sorting signals. The Golgi adds specific signals or tags to proteins that determine their final destination within or outside the cell. These signals guide vesicles carrying the proteins to the appropriate cellular compartments. 6. Quality control. The Golgi apparatus acts as a quality control center. It checks the proper folding and modification of proteins, ensuring that only correctly processed proteins are sent to their designated locations. In summary, the Golgi apparatus is a central hub for protein processing, modifying, sorting, and packaging. Its functions are vital for the proper functioning of the cell, as it ensures that proteins are correctly processed and directed to their intended cellular destinations. 5a. Describe the role of DNA polymerase in DNA solution. Solution. DNA polymerase is a crucial enzyme in DNA replication, responsible for synthesizing a new strand of DNA during cell division. It adds nucleotides to the growing DNA chain in a 5'3" direction, using the existing template strand as a guide. DNA polymerase ensures accurate and efficient replication by proofreading its work, correcting any errors in the newly synthesized strand. It also requires a primer, usually composed of RNA, to initiate the synthesis process. Overall, DNA polymerase plays a key role in maintaining the integrity and fidelity of genetic information during DNA replication. The primary role of DNA polymerases is to accurately and efficiently replicate the genome in order to ensure the maintenance of the genetic information and its faithful transmission through generations. This is not a simple task, considering the size of the genome and its constant exposure to endogenous and environmental DNA-damaging agents. Thus, a number of DNA repair pathways operate in cells to protect the integrity of the genome. In addition to their role in replication, DNA polymerases play a central role in most of these pathways. While polymerases in bacteria, yeast, and mammalian cells have been extensively studied, much less is known about their counterparts in plants. ADP is considered the energy currency of a cell. Justify this statement. Solution. Adenosine triphosphate, ADP, serves as the energy currency of a cell, due to its role in storing and transferring energy within biological systems. Through ADP hydrolysis, energy is released, providing the necessary fuel for various cellular processes such as metabolism, muscle contraction, and active transport. This ability to release and transfer energy makes ADP crucial for powering the diverse activities essential for cellular function. 1. Energy storage. ADP stores energy in its high-energy phosphate bonds, making it a compact and readily available source of potential energy within the cell. 2. Universal energy carrier. ADP is utilized across different cellular processes as a common energy carrier, ensuring compatibility and efficiency in energy transfer. 3. Hydrolysis releases energy. The hydrolysis of ADP into ADP, adenosine diphosphate, and inorganic phosphate releases energy, providing an immediate and controlled source of energy for cellular activities. 4. Chemical versatility. ADP's structure allows it to participate in various biochemical reactions, facilitating energy transfer and coupling with diverse metabolic pathways. 5. Quick energy transfer. ADP can rapidly release energy upon demand, enabling quick responses to cellular needs, such as muscle contraction or active transport across cell membranes. 6. Regeneration potential. ADP can be rephosphorylated to regenerate ADP, ensuring a continuous and sustainable supply of energy for ongoing cellular functions. 6a. Describe electrophoresis as a technique for the separation of amino acids. Solution. Electrophoresis is a technique used to separate molecules based on their charge and size in an electric field. When applied to amino acids, the process involves the following steps. 1. Preparation of gel. A gel matrix, commonly agarose or polyacrylamide, is prepared. This gel provides a porous medium through which molecules can migrate during electrophoresis. 2. Sample loading. The mixture of amino acids is applied to the gel. To ensure separation based on charge, the sample may be treated with a negatively charged detergent to neutralize the inherent positive charge of amino acids. 3. Application of electric field. An electric field is applied across the gel. Since amino acids carry different charges due to their varying side chains, they move through the gel at different rates according to their charge-to-mass ratio. 4. Migration of amino acids. Negatively charged amino acids migrate toward the positive electrode, while positively charged ones move toward the negative electrode. The speed of migration is influenced by the size and charge of the molecules. 5. Separation based on charge. Amino acids separate based on their charge differences, creating distinct bands or spots along the gel. The gel can be stained to visualize these separated amino acids. 6. Detection and analysis. After electrophoresis, the separated amino acids can be detected and quantified using various techniques such as chromatography or specific staining methods, allowing for further analysis and characterization. B. Discuss any one method used for sequencing of amino acids. Solution. One commonly used method for sequencing amino acids is Edmond degradation. Here's an overview of the Edmond degradation process. 1. Derivatization of amino terminal residue. The method begins by selectively reacting with the amino terminal residue of the polypeptide or protein. This reaction results in the conversion of the amino terminal residue into a stable, labeled derivative, typically phenylthiohydantoin, PTH. 2. Cleavage of labeled residue. After derivatization, the labeled amino acid is cleaved from the polypeptide chain without affecting the rest of the sequence. 3. Separation and identification. The labeled amino acid derivative, PTH amino acid, is separated from the reaction mixture and identified using chromatographic techniques, often high-performance liquid chromatography, HPLC. 4. Repetition of the process. The process is then repeated on the newly exposed amino terminal residue of the truncated peptide. The cycle can be iteratively applied to sequence the entire polypeptide. 5. Advantages. Edmond degradation is known for its accuracy and reliability in determining the sequence of amino acids. It is suitable for relatively short polypeptides due to the repeated nature of the degradation steps. 6. Limitations. The main limitation of Edmond degradation is that it becomes less efficient with larger proteins due to the accumulation of errors in the repetitive cycles. High-throughput sequencing methods, such as mass spectrometry-based techniques, have largely replaced Edmond degradation for large-scale protein sequencing. 7a. Differentiate between anabolism and catabolism. Solution. Anabolism and catabolism are two complementary processes in cellular metabolism that involve the synthesis and breakdown of molecules, respectively. Here's the differentiation between the two. 1. Definition. Anabolism. It is the set of metabolic pathways that build larger molecules from smaller ones, requiring energy input. Catabolism. It involves metabolic pathways that break down larger molecules into smaller ones, releasing energy. 2. Purpose. Anabolism. The primary purpose of anabolism is to construct complex molecules, such as proteins and nucleic acids, needed for cellular structure and function. Catabolism. Catabolic processes provide energy by breaking down complex molecules into simpler ones. This energy is often utilized for cellular work and to maintain metabolic activities. 3. Energy requirement. Anabolism. It is an energy-consuming process, requiring an input of energy to drive the synthesis of larger molecules. Catabolism. Catabolic processes release energy as a result of breaking down complex molecules, making it available for cellular activities. 4. Example reactions. Anabolism, protein synthesis, DNA replication, and photosynthesis are examples of anabolic processes. Catabolism, cellular respiration, which involves the breakdown of glucose to produce energy, is a catabolic process. 5. Nature of products. Anabolism, the end products of anabolic reactions are typically large and complex molecules, contributing to cell growth and maintenance. Catabolism, catabolic reactions produce simpler molecules, often in the form of energy-rich compounds like ADP. 6. Overall outcome. Anabolism, it leads to the growth and maintenance of cellular structures and the storage of energy in the form of complex molecules. Catabolism, it provides the necessary energy for cellular activities and is essential for breaking down nutrients to support anabolic processes. In summary, anabolism and catabolism work together to maintain the balance of cellular metabolism, with anabolism building up cellular components and catabolism providing the energy needed for these processes. B. Write the reactions of conversion of glyceraldehyde 3-phosphate to pyruvate during glycolysis. Solution. The conversion of glyceraldehyde 3-phosphate to pyruvate during glycolysis involves a series of enzyme-catalyzed reactions. Here are the key steps. 1. Glyceraldehyde 3-phosphate dehydrogenase, GAPDH, reaction. Glyceraldehyde 3-phosphate, GAP, is oxidized and phosphorylated by NAD, forming 1,3-bisphosphoglycerate. Reaction. Glyceraldehyde 3-phosphate plus NAD plus pi 1,3-bisphosphoglycerate plus NADH plus H. 2. Phosphoglycerate kinase reaction. 1,3-bisphosphoglycerate donates a phosphate group to ADP, generating ATP and 3-phosphoglycerate. Reaction. 1,3-bisphosphoglycerate plus ADP 3-phosphoglycerate plus ADP. 3. Phosphoglycerate mutase reaction. 3-phosphoglycerate is isomerized to 2-phosphoglycerate. Reaction. 3-phosphoglycerate 2-phosphoglycerate. 4. Enolase reaction. Water is removed from 2-phosphoglycerate, forming phosphoenolpyruvate, PEP. Reaction. 2-phosphoglycerate phosphoenolpyruvate plus H2O. 5. Pyruvate kinase reaction. Phosphoenolpyruvate donates a phosphate group to ADP, producing ATP and pyruvate. Reaction. Phosphoenolpyruvate plus ADP pyruvate plus ADP. These reactions collectively represent the conversion of glyceraldehyde 3-phosphate to pyruvate in the latter stages of glycolysis. The process results in the net production of ADP and NADH, contributing to the overall energy yield of glycolysis.